Hello. Thank you so much for inviting me to come here and talk. This has been an awesome day of presentations, so I hope I don't drop the ball. I'm from City of Hope, which is a cancer center outside of Los Angeles. We have a 30-year-long graduate program where we train PhDs. We work very closely with our clinician colleagues. My lab in particular is trying to understand why older women get more breast cancer. So, in fact, all cancer is more or less age-related. And this is a figure from James DeGregory's recent paper, or recent-ish paper. And you can see that even up here, when you're looking at BCR-ABL induced CML, it's just one translocation, still shows this interesting relationship, incidence and age, okay? If you normalize all of these different kinds of cancers, you have heme and leukemias up here, you have different solid cancers down here. When you normalize those as normalized incidence, you see that age is the dominant factor. And the point you make from this, or that we make from this, is that aging and the relationship between aging and cancer is you become more susceptible to cancer as you get older, okay? And why is that? This BCR-ABL thing, I think, is a very interesting example, and it flies in the face of the notion that we have a multi-hit hypothesis. And I just want to leave this in the back of your minds, that the multi-hit hypothesis is probably wildly overstated. So, zooming in a little bit more to breast cancer. Over on the left is a list of things that are risk factors for breast cancer. On the bottom, where you have relative risk less than twofold, that group of risk factors represents the dominant numbers of publications of people trying to understand risk factors for breast cancer. And I think that's because those are all things that we can control or try to control. Okay, the fact of the matter is their impact on breast cancer risk is very, very modest, okay? It's not until you start working your way up the list that you start to see what I think are the real drivers of breast cancer incidence: inherited risk, like BRCA1 genes, BRCA2 genes, atypical hyperplasia diagnosis, and age is the dominant factor. Over on the right is a distribution of how old women are when they get breast cancer in the United States. And you have this, I think, quite satisfying double mode where you have one group of women who are sort of in their 50s, and you have a group of women sort of in their 70s, and that's where you're getting the peaks of breast cancer incidence. This isn't incidence, this is density, okay, just to be clear. If you look at the same distribution in women who live in Japan, it's totally different, okay? Now, not only is frequency lower, okay, so there's less breast cancer, but it is a disease of younger women, right? Now, what's interesting is that when women relocate from Japan to the United States in their teens and 20s, this happens. And they start to, the Japanese women start to sort of, at the level of breast cancer and the age at which it's being diagnosed, start to look more like the American women. And the first time that I saw these data, I thought like, wow, okay, that's what I'm gonna work on the rest of my life, because this second hump is modifiable. That is modifiable risk. It's some combination of aging and environment that is causing the majority of breast cancer, and that is modifiable risk, because it doesn't have to be there, okay? What we have to understand is why it's there. Why are this group of older women susceptible to cancer as they age? So, when I'm talking to you for the rest of this talk, until the very bitter end, I'm not talking about studying cancer cells. In order, because cancer, we say in the United States, the horse has already left the barn. All the bad stuff that's gonna happen has happened, and now we have to like regroup and think about how to study this phenomenon. So we have to study normal, and we have to understand sort of the distribution and what defines normal, okay? Can't do this in most model organisms that are convenient for aging. A lot of them lack the relevant organs, and even those that have mammary glands, like mice, which is, seems to be the favorite organism, they actually, wild-type mice do not get age-related mammary tumors, number one. Number two, the genetically modified animals get the tumors very early, and they do not mimic breast tumors that we see in women, in older women. Okay, so this wasn't a good choice for me to understand this problem that I want to go after. Now, in aging, by and large, when we're sampling from humans, we're looking at tissues that are not representative of the epithelial tissues, which represent the vast majority of age-related cancers, carcinomas. We're looking at things like urine, buccal cells, blood, skin, feces, things that are easy to get, and it's understandable why that should be the case, okay? However, there's an interesting opportunity with breast tissue because, let me see, there we go. Because of prophylactic surgeries and cosmetic surgeries, breast tissue is a fairly easy to access, discarded breast tissue is a fairly easy to access tissue that can be normal, okay? And so for many years, actually my colleague started this bank, Martha Stanford, back in 1974 when the year I was born, and she'd collected a few hundred reduction mammoplasties. Having joined her and worked with her for many years, I added to that, and now we've added several hundred samples from our clinicians at City of Hope, where we've not only gotten genetically normal breast tissue, but a lot of tissue from our genetic testing unit that have BRCA1 mutations, BRCA2 mutations, and lots of other mutations that are relevant to breast cancer risk. We have an excellent cell culture system. We can take epithelia, fibroblasts, immune cells, adipose. We can mix them back together, do multi-tissue, multi-lineage co-cultures. The cell culture technology that we've developed will take these epithelial cells out 60 population doublings or more, so that's about 15 passages, as primary cells. These are not genetically manipulated in any way, okay, and they're still finite. And basically, at fourth passage, which is about 12 population doublings, is where we do most of our work. The gang is all there. We've published so many papers proving to people that the transcription is the same, the proteome is the same, the methylome is the same. It's really an excellent model, and I would invite you to join us in our journey. This is where the cells come from, and I just wanted to introduce. For those of you who don't know about the mammary gland, this is an up close and personal cross-section. So, oops, there we go. Right in the duct, in the middle, this is the terminal duct. These orange cells are stem cells, which give rise to the green secretory cells, luminal epithelial cells, and to the red myoepithelial cells, which are contractile and tumor suppressive, okay? I have been, for probably 20 years, trying to understand how the external milieu, the microenvironment, imposes itself on these different cell types, both to exact differentiation in sulfates, but also maintain those sulfates. And I think a big part of aging and susceptibility involves those mechanisms that maintain sulfates going awry. So, since I've had a lab using this system, what we have discovered is basically the following. This is a brief summary. So, number one, the tumor suppressive myoepithelial cells proportionately decrease with age, okay? You're accumulating dysfunctional multipotent progenitor cells. So they stop actually answering to normal cues in biology, or normal biological cues for differentiation. There are changes in the molecules that maintain bilayered organization, okay? So you can already see how this is going towards what cancer might look like. Telomeres don't shorten. There is a relative difference between the luminal and the myoepithelial cells in every person, but as a function of age, we don't see convincing evidence of shortening. So that's not a thing here. Luminal cells look really, really basal-like with age. They lose their lineage specificity. And I think this is a very, very important aspect, a hallmark of aging in breast. And then finally, when we look out at the immune milieu, as women get older, the immune milieu becomes more immunosuppressive, and you see more senescence in the CD8 positive T cells. So, moving to City of Hope was really important for me because we have such an aggressive genetic testing unit. We were able to collect a lot of tissue from women who had BRCA1 mutations and other mutations that I talked about. They're an important group because they are 80% likely in their lifetime to have a cancer, whereas average risk in the United States is about 1 in 13, 1 in 12, okay? So we needed a group of women who we were sure was gonna get cancer because we wanted to know if these hallmarks of aging that we'd been describing had anything to do with risk. And these two rows on one side on the left is just plain old aging, and on the right are young women with these high-risk mutations. And by and large, we actually see most of the things that we were calling aging happening 20, 30, 40 years earlier in women who were high risk. So these aren't just aging things that I'm throwing out, they are directly related to risk, okay? One of the first things I saw, I'm just gonna walk you through a couple of these examples. One of the first things we saw were changes in the basal keratin expression. So here's a young, healthy duct where you see the green keratin 19 and red keratin 14. This is sort of what the markers should look like in a young person. As women get older, the luminal cells start to pile up, they're losing organization, and they start to express this basal keratin 14. And young women with BRCA mutations, that happens much, much faster. The luminal cells, when we culture them in a dish from young women, they hate being on a dish. They cannot interact with matrix to save their own life. But you put luminal cells from an older woman or a woman with a BRCA mutation, and they're in hog heaven. They go nuts. So there's a completely new apparatus coming online as we age during this loss of fidelity that is essential to cells communicating with ECM. And we were very, I think, astonished to discover that. And then this is an example, we talked earlier about biological clocks. This is a very simple biological clock that's breast specific. You do need a little bit of breast tissue in order to do this. My point is, this is accurate to within about 2 to 3 years to predict how old a person is chronologically. And it's based on the expression of a single gene, ELF5, which is a transcription factor that maintains the luminal epithelial cell fate, okay? If you intimately understand the biology of the tissue that you want to understand in the context of aging, I think you get a lot more options in terms of biological clocks and so forth. Interestingly, this allows us to do a little math, and basically women who are in this high risk group, and it doesn't matter which of the inherited mutations we've looked at, they all show the same phenotype, and we can calculate based on this sort of simple regression that they're 10 to 40 years older than their chronological age. We think this might be an interesting way of screening women for assessing risk. So, if you haven't been paying attention, I'm going to bring you up to speed. The majority of the changes that we see with aging, like the real hallmark changes, the obvious ones that hit you in the face, are happening in the luminal epithelial cells. And this merits our attention because that is a group of cells that is essentially the cell of origin for age-related breast cancer. That's where all the action is happening, and it's probably not by accident, okay? So we've really taken a deep dive into understanding this. Rosalind Simon, when she was a postdoc with me, took the first sort of genome-wide look, and what you see here are genes that are differentially expressed in luminal cells or myoepithelial cells with age, or differentially methylated CpG islands in luminal or myoepithelial cells. And all the change is happening in luminal cells, right? You have around 734 changes in gene expression, and you have around 22,000 differentially methylated CPGs. Almost no change in the myoepithelial cells, right? These come from the same progenitor. They live next to each other in the tissue. Oh my God, I'm going to go way faster. Okay, so what they do have in common is that both lineages show increased variability. So there's more noise in the transcriptional system as women get older, okay? There's a member of our audience, Parajat Senapati, who I got to work with for a few years on this project. We're trying to understand the sources of the variance. One of the sources of the variance is that evolutionarily young transposable elements become demethylated as women age, just their luminal cells, by the way, which starts sort of wreaking havoc in the transcriptome, turning on things that aren't supposed to be turned on, okay? Another source is sort of stochastic variance around genic promoters. On the left in this inset is all of the CPGs, genic CPGs, and ordered from highest methylation to lowest, and then the corresponding promoter in our group of older women. And what you see in this larger one here is where all the variances is. And all the variance comes in the hyper and hypomethylated regions of the promoters. And what's especially interesting is where we see increases in methylation. It's happening at the luminal specific promoters. So those are getting shut down. And where we see decreases, it's happening at the myoepithelial specific promoters. So right before our eyes, these cells are doing a switcheroo. And it's all because of this increased variance. Now, we know the myoepithelial cells are changing. And actually, we did a heterochronic co-culture where we make feeder layers of young or old myoepithelial cells and culture young luminal cells on top. And when we do young and young, we get young gene expression. And when we put young luminal cells, are you sure? When we put young luminal cells on top of old myoepithelial cells, their transcription converts to that of an older cell. And this is important because those changes in just gene expression variance in the myoeps is enough to exert directional changes in the luminal cells, okay? Now, I'm gonna go through this next part quick, but this is really important because what we're starting to see, the pattern that's emerging, is there's a group of CPG islands that has heretofore been ascribed to breast cancer, that they were breast cancer specific differential methylation. We see all that happening in normal, older luminal cells. And what tends to be the case is an example here by this, it's a gene called CXXC5, which is a zinc finger that binds unmethylated DNAs. We see that with aging, you lose methylation, you have a change in methylation, and a corresponding change in transcription that tends not to be significant. And then when you look in the cancer, those changes tend to be statistically significant. And we dug a little deeper on this gene called CXXC5. Joe Casano did, and he confirmed that the changes in transcription correspond to changes in protein. Most of that change happens in the nuclei of older luminal cells. Now we have a nice way of pushing our normal cells down the malignant path. And as you sort of bypass the stress-associated senescence by overexpressing cyclin D1, and then overexpressing c-MYC to bypass replicative senescence and make them immortal, when you hit that immortal stage, you see that CXXC5 goes through the roof, okay? Now we asked, does CXXC5 actually drive immortalization? Does it bypass replicative senescence? No. So when we overexpress it, they all just become senescent, okay? But if we now take cells that have already bypassed stress-associated stasis and we knock down CXXC5, most of them become senescent. And so it's telling you that this is not sufficient to immortalize the cells, but it is required. It's making the genome permissive to the state of immortality, which is a rate-limiting step in cancer. This is extremely important. One more. And just sort of to drive that home a little bit, when we looked at CXXC5 expression in the senescent cells, they did not come back up. When we looked at the one clone that immortalized, it had this bump in CXXC5 expression. And we're sort of pursuing this. This is early data, and you're going to see the good one down the road. I'm just going to take about 30 more seconds. I apologize to the other speakers. So this is a paper by the Suderman group. And I wanted to bring this up because they looked at gene expression in pieces of tissue, basically. Okay, not different lineages, pieces of tissue. All the variance was pretty much driven either by the environment or by aging. And the variance that they measured was tissue specific. So the genes that become variant with age are tissue specific sets of genes. So if you want to understand aging and disease, which is tissue specific, you need to understand the tissue of interest, right? So in general, disease is tissue specific, and we should really try to study how aging impacts those tissues directly instead of using proxies. In breast, there's increased variances in DNA methylation transcription that basically result in loss of lineage maintenance, weakens the intrinsic defense mechanisms, and enables bypass of replicative senescence. That was a specific example that I shared. And what my lab is working towards understanding are these different levels of cell-cell communication and how we can control them to either prevent or detect cancer much earlier. And this is my group, and I'm sorry I went over a little bit. So thank you.